In this video, I'm just going to review meiosis 2. Remember, when you think of meiosis 2, this is actually the same thing as mitosis, except for you end with 4 cells, and this happens with 2 cells along the way. So what I would encourage you to do at this time with me, we start with prophase 2. There is no interphase 2. So you're going to start with prophase 2 for meiosis 2, and then you're going to label prophase, metaphase, anaphase, telophase, cytokinesis. I would go down right now and I would label all of those with the 2 after them. And then I would pause the video and I would start drawing your cells and do the same thing as mitosis. Of course, you can stop and double check your answers and your work maybe for prophase and metaphase before you keep going, just to make sure it's right, but it should be the exact same as mitosis. So if you feel comfortable doing that, I would pause the video and start doing that, and then of course you can come back and listen and make sure that you did this correctly. So remember, we had 8 chromosomes, and remember our end result from our last drawing. So what I recommend to do is to always go back to your last drawing, which we had, and telophase right there. I always look at that one before I start my prophase on my prophase 2, because prophase 2 I have to take what I had over on the other side and draw it. Okay, so mine are kind of on opposite sides, but you can see that I have 3 from mom, 1 from dad, 3 from dad, 1 from mom, and that's going to start our prophase. So I'm just going to quickly review this with you. What's going to happen in prophase 2 are the 3 same events that always happen. Remember, the nuclear membranes will disappear, spindle fibers will appear from those centrioles, we'll have chromatin that's going to coil into chromosomes. After we got done with cytokinesis, remember from our cytokinesis 1, it went back to its relaxed state, so now it has to coil back up in order to go through a second division. So prophase 2 only has those 3 main events, just like normal. Then we go to metaphase. This one looks different than metaphase 1 because they line up single file. This is just like our mitosis metaphase because they line up single file instead of with buddies. Remember, in metaphase 1, they found their buddy and that had the same genes in the same locations, and they buddied up and they were kind of in pairs in the middle of the cell. So the only thing that sometimes goes wrong with metaphase is you need to take these guys and put them in the middle, and take those guys, make sure you color code them, and you put those in the middle. And you can put them wherever you want. I could have put the mom chromosome up there and the dad ones down there, or vice versa. It doesn't really matter where you put them. It's completely up to you. But they do have to be single file. So duplicated chromosomes lining up single file in the middle of the cell. Remember, metaphase, middle. Now, after we went through prophase 2, metaphase 2, we go to anaphase 2. With anaphase 2, this is where, looks just like mitosis again. We separate the sister chromatids. So always look up here. This is the mom one, so you got to separate the sister chromatids. Those are from mom, those are from mom, just like up there. So we had 1, 2, 3, 4 duplicated. So we still have those 4, but they're split now. So we have 8 unduplicated chromosomes here. You can see how we're getting our numbers now. Same thing here, pay attention to the color coding. They're all going to split, so be careful with how you put them to the sides. You can see they're looking a little bit different. So sister chromatids are going to separate in anaphase, just like in mitosis. And then we go to telophase. In telophase, this is where you really got to pay attention to what happened in anaphase. Think this side right here is going to go right there. Okay, this side right here is going to go on the other half of that cell. This part right here is going to go right there. And this part right here is going to go over here. So think of those cells splitting in half. That's exactly what's happening. So think of drawing a line in the middle, and think you're getting the nuclear membranes again reforming around those chromosomes. The spindle fibers are disappearing, and then those chromosomes are going to unwind and form chromatin again. Okay, so that's all happening in telophase. You can tell now we only have 1, 2, 3, 4 unduplicated chromosomes finally. Remember, we ended meiosis 1 with 4 duplicated chromosomes. Now we have unduplicated ones. So what do we end with? Lastly, cytokinesis. Remember, with cytokinesis, we're going to split the cytoplasm. I don't even think I wrote that, but that's what's going to happen in cytokinesis 2. We're going to split the cytoplasm just like usual, but it's going to happen with 2 sets of cells. we're going to end up with 4 haploid cells. Remember, you get sex cells from this whole process after the second division here. They can also be called gametes. In males, these are going to be sperm. They're all going to be approximately the same size. In females, you're going to get 1 egg usually. The other 3 are called polar bodies. And the egg is usually much larger, and the polar bodies are just teeny tiny little ones. They usually just kind of disintegrate, and they're not used as eggs. So in a human, what do we get inside of here? We would get 23 chromosomes in each of our cells, which is called a haploid cell. Remember, we would start with 46 chromosomes at the very beginning. For this one, we started with 8. So technically, this one, we have 4 unduplicated chromosomes in each one of our final cells here. So in our sperm or egg at the very end. So recap, remember meiosis has 2 divisions. Remember, the whole purpose is to produce sex cells, which are gametes or sperm or egg, and this process only happens in the testes and ovaries of organisms. So hopefully you feel a little bit better. If you need to recap anything, of course you can stop the video, go back and look at anything that you need to kind of revisit. Thanks for watching.